Have you ever wanted to understand, say, a whole university course, but, skip the lectures and just get the really fascinating parts? Well, that's pretty much our mission today. We're taking a deep dive into plant biotechnology, specifically how it's really changing plant breeding. And our insights for this, they come straight from the source, the introductory materials for a real university course, FIT 679.
Oh, yeah. That's taught at the Federal University of Viçosa, UFV, in Brazil. Professor Guilherme da Silva Pereira.
Exactly. And our goal here is to pull out the core ideas, and we're going to focus on a really great real-world example, genomics-assisted breeding in sweet potato. So get ready for maybe some aha moments, maybe some surprising facts, and hopefully a clearer picture of how the science is changing our food. Okay, so picture this. You're signing up for this advanced course on plant biotechnology, but hey, we've got a bit of a shortcut for you right here. This course, it's pretty intensive, 15 weeks, 60 hours.
Right. Offered by the Department of Agronomy at UFV. It's got Professor Pereira leading, but also guest lecturers, teaching assistants. It's the full university setup.
And a key thing for the students, which is relevant for us, is this final project. They have to pick a crop, find a breeding gap, like maybe it needs better stress tolerance or better quality or more yield, and then propose a biotechnology approach to fix it. And that's kind of what we're exploring today using that sweet potato example.
And what's really neat about that structure is how it immediately focuses on solving actual problems.
Yeah.
It's not just abstract theory from day one. It's about application.
Yeah, it gets right into it.
The course outline itself, it builds logically.
Yeah.
Starts with the basics, the molecular tools markers, PCR, S&Ps.
The toolkit.
Exactly, the toolkit. Then it moves into how you use that toolkit.
Right.
Pre-breeding, looking at genetic diversity, mapping genes.
QTL mapping, genomic selection.
All the way up to gene discovery, molecular breeding, transgenics, and even, the really cutting-edge stuff like CRISPR-Cas9 genome editing, plus biosafety considerations. It covers the whole spectrum.
Which brings us to where Professor Pereira really connects the dots. Sweet potato. He uses it as this prime example for applying all these biotech tools. And honestly, it's way more impactful than you might first think.
Oh, absolutely.
I mean, sweet potato is a massive deal, right? A vital root crop, especially in Latin America, Africa. China's the biggest producer globally.
Huge staple food.
But there's this key difference, isn't there, between the white flesh types common in Brazil, Africa, Asia, and the orange-fleshed ones you see more in, say, Europe or the U.S.
Right. And that color difference is critical.
Why is it so critical?
Well, that orange flesh, it's packed with beta-carotene, which the body converts to vitamin A.
Ah, vitamin A. So we're talking nutrition.
Exactly. We're talking about fighting hidden hunger, that micronutrient deficiency. It's huge for infants, pregnant women. It's basically biofortification right there in the crop, which is vital for smallholder farmers.
So science meeting a really fundamental human need.
Precisely. But the challenge, and this is where the breeding comes in, is that many of those desirable orange flesh varieties were developed, up north, adapting them to African conditions. That takes a lot of work.
Different climates, different pests.
Exactly. And that's where biotechnology, specifically genomics, can really speed things up massively. In fact, there was a project focusing on orange flesh sweet potato that won a World Food Prize back around 2012.
Yeah.
Shows you the scale of the impact.
Wow, okay. But sweet potato itself, it has this genetic complexity, right? It's not straightforward.
Not at all. It throws a real curveball.
The cultivated kind, Ipomoea batatas, it's a hexaploid. What does that actually mean, hexaploid?
Okay, so think about humans, or many common plants. We're diploid. 2 sets of chromosomes, one from mom, one from dad.
Right.
Sweet potato, it's got 6 sets. 6. 2n equals 6x equals 90 chromosomes. Its wild relatives are diploid, just 30 chromosomes. This polyploidy, having extra sets, often leads to bigger plant parts, like those nice big sweet potato roots.
Which is good for yield.
Good for yield, yes. But, and this is the big but, why does this ploidy level matter so much for breeding?
Yeah, why is 6 sets harder than 2?
Because it makes the genetics incredibly complicated. With 6 sets, you have multiple homologous chromosomes that are similar, and they can pair up in all sorts of ways during meiosis when sex cells are formed.
Okay.
Tracking which gene version goes where, it becomes a statistical nightmare compared to diploids. And sweet potato isn't alone here. Sugarcane, strawberries, wheat, lots of important crops are polyploids.
Different kinds, though.
Yeah, there are autopolyploids like sweet potato and potato, where the genome basically duplicated from the same species. This leads to really complex chromosome pairing called polysomic inheritance. Then there are allopolyploids from hybrids of different species, which often behave a bit more simply, more like diploids. Sweet potato is an autopolyploid, the complicated kind.
A real genetic puzzle. So how on earth do scientists even start to unravel that complexity to actually breed better sweet potatoes faster?
That's where the molecular tools become absolutely essential.
Mm.
We need markers. Specifically, SNPs, single nucleotide polymorphisms, those tiny variations in the DNA sequence.
Right, SNPs. But how does that work in a hexaploid? If a diploid usually has two options, A or T, what about six sets?
Exactly. In a diploid, SNP is usually biallelic, two versions. In a hexaploid like sweet potato, at one spot, you could have 0 copies of allele A, or 1 or 2, 3, 4, 5, or 6 copies. This is called allele dosage.
Okay, 0 to 6. That's a lot more possibilities.
It explodes the complexity. Think about it. You cross 2 hexaploid sweet potato parents. Theoretically, the number of different genetic combinations, genotypes, in their offspring could be up to 400.
400. You can't possibly grow out and check 400 combinations for every cross in the field.
Absolutely not. It's impossible, which is why we need high-throughput methods.
Like genotyping by sequencing, GBS.
Precisely. GBS has been a game changer, especially for complex polyploids.
So how does GBS manage to cut through all that complexity? What's the trick?
It's clever. First, it uses restriction enzymes, think of them as molecular scissors, to chop up the DNA, but only at specific recognition sites. This creates a smaller, manageable subset of the whole genome.
Okay, makes it less overwhelming.
Right. Then, it adds unique DNA barcodes to the DNA fragments from each individual plant sample.
Barcodes, like in a supermarket?
Sort of. It
Fair.
means you can pool hundreds, even thousands of samples together and sequence them all in one go on a next-generation sequencer. Super cost-effective.
Ah, multiplexing. Got it.
Then the bioinformatics pipelines take over. They sort the data using those barcodes, identify the SNPs, and crucially for polyploids, they can estimate that SNP dosage, the 0 to 6 copy number, based on how many times they read each allele version.
So it discovers the variations and genotypes them, including the dosage, all at once.
Exactly. You don't need prior sequence information. It finds the SNPs and counts the alleles simultaneously. It was a massive leap forward. Tools like GBS poly, Tassel4 poly, MAP poly, QTL poly. These were developed specifically to handle this polyploid data from GBS.
Okay, wow. So you get this huge amount of SNP dosage data. What's the next step? How do you make sense of it?
The next step is building the linkage map.
The genetic roadmap.
That's a good way to put it. You use statistical methods to figure out which markers tend to be inherited together because they're physically close on the same chromosome. You estimate the recombination frequency between them, group them into linkage groups corresponding to chromosomes, order the markers within each group.
And figure out which parent contributed which version.
Right. That's called phasing the haplotypes, determining which block of variants came from which parental chromosome.
And this has actually been done for sweet potato.
Oh, yes. There's a key study using a cross between Beauregard, that's a major orange-fleshed U.S. variety, and Tanzania, a white-fleshed African one.
Okay.
They generated about 315 offspring, full siblings,
Yeah.
and they genotyped them using GBS, getting data on over 1 million SNPs.
1 million?
Yeah. From that, they built a high-density linkage map with 15 linkage groups, which perfectly matches the 15 chromosomes sweet potato has.
So the map worked.
Beautifully. It shows you the order of genes, the distances, and you can even see evidence of that complex chromosome pairing multivalent formation that happens in autopolyploids like sweet potato. You're literally seeing the structure of inheritance.
Incredible. So, okay, we have the map. We know where things are, generally. But how do you find the specific locations, the treasures, as you called them, the genes that actually control the traits we care about, like that beta-carotene or yield?
That's the goal of QTL mapping, quantitative trait loci mapping.
Right. QTLs.
So you have your mapped population, like the Beauregard X Tanzania cross.
Mm-hmm.
You've measured their traits, their phenotypes, how much beta-carotene, how much dry matter, what's the yield.
The observable stuff.
Exactly. Then you look for statistical associations. Are individuals that inherited a specific chunk of chromosome from, say, the Beauregard parent also consistently higher in beta-carotene? If you find such a region, that's a QTL. It's a region of the genome linked to variation in that quantitative trait.
And did they find QTLs in that sweet potato study?
They certainly did. For the quality traits, dry matter, starch, beta-carotene, flesh color, they found they were largely oligogenic.
Oligogenic, meaning?
Meaning controlled by just a few genes, or QTLs, that have relatively large effects. For example, they found 5 main QTLs for dry matter that together explained over 67% of the variation in that trait. That's huge.
Wow. And beta-carotene. The orange color.
Yes, crucially, they found major QTLs for both beta-carotene and starch content located very close together, collocated, on chromosome 3 and also on chromosome 12.
Close together. Does that explain why sometimes high beta-carotene varieties have lower dry matter or vice versa, that negative correlation people observe?
That's exactly what the mapping helped clarify. On chromosome 3, they found that the QTL peak for beta-carotene was right near a known gene, phytoene synthase, involved in making carotenoids. And the starch QTL peak was right near sucrose synthase, involved in making starch.
So two different genes right next to each other.
Exactly. It's linkage. The genes are physically close on the chromosome and tend to be inherited together. It's not pleiotropy, where one gene affects both traits.
Why is that distinction important, linkage versus pleiotropy?
It's really important for breeding. If it were pleiotropy, improving one trait might automatically hurt the other. But because it's linkage, there's a chance through recombination over generations, or maybe even using gene editing, to break that linkage, to get high beta-carotene and high dry matter starch in the same variety.
Oh, okay. That's actually good news then.
It is. And on chromosome 12, they found another major player, the orange gene. This gene is known to control the formation of chromoplasts, the cellular structures where carotenoids accumulate.
The orange gene. Does that explain how some offspring can be even more orange than the orange parent, that transgressive segregation thing?
Precisely. Offspring can inherit favorable alleles, like a functional orange gene allele from both parents, potentially getting a higher dose of orange-promoting genetics than either parent had alone, leading to super orange progeny.
Fascinating. What about yield traits, though? Were they also controlled by just a few big QTLs?
No, and that's typical. Yield-related traits, like root yield itself, tend to be polygenic.
Polygenic. Many genes involved.
Many genes, each with small effects, and often interacting strongly with the environment. This makes finding major, stable QTLs for yield much, much harder. The genetic control is just more diffuse.
Okay, so we have oligogenic traits with big QTLs, like beta-carotene, and polygenic traits like yield with smaller, numerous effects. What does this mean practically? How does all this knowledge turn into better sweet potatoes on our plates?
It leads directly to smarter breeding strategies. Genomics-assisted breeding.
Right, making breeding faster and more precise.
Exactly. For those oligogenic traits where you found major QTLs like beta-carotene, or they also found a big QTL's resistance to root knot nematodes, Maloidogyne incognita, on chromosome 7. You can use marker-assisted selection, or MAS. You develop simple assays for the SMT markers right next to or inside the key gene or QTL. Then you can screen thousands of seedlings in the lab using just a tiny bit of leaf tissue.
Like a genetic shortcut, the cheat sheet.
That's a perfect way to put it. You select the seedlings with the desired marker allele combination without having to wait years to grow them out and measure the trait, especially for things like disease resistance, which can be hard to screen reliably in the field. Much faster, much more efficient.
Okay, that makes sense for the big effect traits. But what about yield, the polygenic ones? Can you use MAS for that?
Not easily, because there isn't one big marker to select for. That's where genomic selection, or GS, comes in.
Genomic selection, is
Right.
that different?
With GS, you don't rely on finding specific QTLs. Instead, you use genome-wide marker data, maybe thousands or tens of thousands of SNPs covering the whole genome.
Yeah.
You build a statistical model in a training population where you do have both genotype and phenotype data. This model learns the cumulative effect of all those markers across the entire genome on the trait, like yield. Then, you can genotype new breeding lines, plug their marker data into the model, and it predicts their likely performance, their genomic estimated breeding value, without you having to phenotype them extensively first.
So it predicts overall genetic merit based on the whole genome pattern.
Pretty much.
Yeah.
It captures all those small effect genes that MAS would miss. It's powerful for complex polygenic traits.
So how do breeders put MAS and GS together for a crop like sweet potato, which has both kinds of traits?
They use an integrated strategy, which is really smart, especially for polyploids. In the early stages of breeding, when you have thousands of potential plants, maybe from many different crosses, you use relatively cheap, maybe fewer markers, focus on those major MAS targets,
Yeah.
screen for high beta-carotene, screen for nematode resistance, get rid of the obvious poor performers quickly and cheaply.
Okay, thin the herd first using MAS.
Exactly. Then you take the hundreds of individuals that pass that first screen. Now you invest a bit more. Genotype them with a denser SNP panel, maybe several thousand markers.
Right.
And you apply genomic prediction using models like GBLUP to predict their performance for the complex traits like root yield, maybe storage root quality, things like that.
So a two-state approach. MAS for the easy wins, GS for the complex stuff.
Precisely. This lets you identify the elite candidates, the ones that have the best combination of all the desired traits, much earlier in the breeding cycle. You shorten the whole process, sometimes by years, which means you get faster genetic gain, better varieties out to farmers faster.
Oh, we've really covered some ground today, haven't we? It's quite a journey from, a university course outline in Brazil deep into the genetic code of the sweet potato
Yeah.
and seeing how that connects directly to tackling big global challenges like nutrition in Africa. It's just amazing to see science having that kind of impact.
It really is. And this whole deep dive into plant biotechnology, using sweet potato as the lens, it just highlights how crucial understanding the genome is, especially for these complex polyploids. It's not just academic curiosity. It's absolutely foundational if we want to engineer the crops we need for the future.
Yeah, making them more resilient, more productive, more nutritious.
For everyone.
So maybe the next time you bite into a sweet potato, orange or white, just consider that incredible journey its DNA has been on and all the scientific ingenuity it takes to make it better, more abundant. It makes you think, what other breeding gaps out there could plant biotechnology help us fill next to shape the future of our food?